Thank you very much. I have a long list of Melanie's accomplishments here, which I'm going to ignore because I've known Melanie for many, many years. And it's a great pleasure to introduce her to you tonight. Melanie received her PhD from the University of London, working in the British Museum of Natural History. We overlapped a little bit at that time when I was doing some research there myself. And after that, Melanie came to Harvard, where she worked as curator of fishes and professor of organismic and evolutionary biology here for almost 5 years before moving to the American Museum of Natural History, where she's now Axelrod curator. She's involved in the graduate program there with Columbia University. She's become widely known for her work on the evolution of fishes, and she's, I believe, chair of the fish department there at the American Museum of Natural History. Melanie's research has just spanned so many different aspects of fish biology, and including studies of their structure, their evolution, focusing for some time on cichlid fishes, very well-known group of an evolutionary test case. But more recently, she's expanded her work out into major field efforts, such as you're going to hear about tonight, the remarkable situation she's uncovered in the Congo River. And it's actually, when I teach biology of fishes, usually in the every other year in the spring here at Harvard, I use her material from this case study to introduce the undergraduate students to an amazing evolutionary microcosm that really integrates geology, ecology, behavior, and evolutionary diversification of fishes. It's an amazing system that she really has been the sole person responsible for developing and pulling a huge team together to address this. So with great pleasure, I'm going to now introduce Melanie Stiassny of the American Museum of Natural History in New York, who will tell you about evolution and diversification in Congo River fishes.
Thank you. Well, thank you so much, George, for that lovely introduction. It really is a pleasure to be here and to have a chance to talk with you a little bit about, as George said, some of the work that I've been doing recently. Well, I say recently, but my first slide is going to show you this cartoon. I say recently, it feels like I've been in the Congo since 1884, like these gentlemen. But in fact, my work there really began in about 2006. So I've been going there for a long time now. And I'm going to kind of start off by, I know we're going to be talking about collections and the importance of collections, but I'm going to start off by talking about my study area, some of the things that I've been finding there, and then I'll segue into the collection side of it and what we actually do with all of these collections that I've been making. So to start off, I'm going to talk a little bit about the Congo Basin. So Africa, I'm sure you're all very familiar with. And the Congo Basin I can slot in there. Now, the Congo is really the freshwater heart of Africa. It's a place of superlatives. It's the world's second largest river basin. It drains some 4 million square kilometers of land in Central Africa. That's about the size of Western Europe. It's a massive drainage basin. The source waters take about 6 months from their source in the southeast to make it to the Atlantic Ocean. And the Congo River also provides about 14,500 kilometers of navigable passage across Central Africa, and it provides the food and livelihoods for about 60 million people who live there. And it's become increasingly important because I know a lot of you you read about the war in the Congo, and most of the drainage of the Congo Basin is actually situated in the country of the Democratic Republic of Congo. Congo's gone through and is still going through some really tough times. And amazingly, today, in a country the size of Western Europe, there are about 300 kilometers of paved road. So the river is an incredibly important component for the peoples of this region, just to get around and to conduct trade and do everything. And my next slide, I don't know if you can see this. It looks like a floating island, right? This is actually a barge. So here you can see the motor of it, and it's moving slowly up the Congo River, carrying all these people and all their goods. So it's a really amazing picture, I think, that kind of gives you a sense of the importance of the Congo. But I'm going to be talking about it as of its biological importance. And to kind of situate what I'm going to be talking about, I've kind of colored in the main stem of the Congo River. So arising here, looping round through the heart of Africa, and actually, it's cut off on this side, but you're actually flowing out into the Atlantic Ocean. Now, I want to draw your attention first just to this little round thing here. And what that is actually a very strange expansion in the middle of the Congo River. It used to be called Stanley Pool, and nowadays it's called Pool Malebo. And the area of the Congo that I'm going to be talking about, sorry, probably I should just go back, is really from that expansion, which I'm going to be talking a little more about, down to the Atlantic River. That small stretch, a tiny percentage of the Congo Basin itself, is what I'm going to be talking about. And in my next slide, I've just kind of highlighted that. So here's this strange swelling. Above it, the middle Congo River flows. Then you come down to this strange swelling, and then the river spills out over a sill and plunges down this lower Congo River to its outflow in the Atlantic Ocean. So that's a short stretch, and I'm going to be talking a lot about it. It's just about 300 kilometers in length. But just quickly to look up here, you can see this strange, very strange kind of expansion in the middle of the Congo River, which is, as I say, was called Stanley Pool, and we now know it as Pool Malebo. It's a very strange structure. We know very little about the geology, the geological history of it. But as far as fishes are concerned, it really forms a biological barrier. The kind of things that you see above the pool, fish-wise, and the things that you see below the pool, fish-wise, are very different. And if I just show you this picture, this is actually a picture taken in the pool. And the pool itself is very shallow, it's very sandy, it's very different from the kind of river that's above the pool, the middle Congo River. And I think that's much more like the kind of African queen that you think about, the big meandering river, the leeches, the flies, the heat. Above the pool, it gets very African queenish. The pool, very shallow, spills out, sandy. And then below the pool, in this study region of mine, the lower Congo River, it's hugely tumultuous, turbulent, high energy, and you're going to see quite a lot about that. So this strange swelling really forms a biogeographical barrier for the fishes above and below, seems to separate them pretty well. Okay, so this is basically what we knew when my work began back in 2006. And this is taken from a paper, and I'm sure you'll recognize the journal, from the bulletin here of the MCZ. And two ichthyologists, Tyson Robertson, Don Stewart, really undertook the first study, the first look at the fishes of the lower Congo River. And this paper was really the first to document that there's something interesting about the lower Congo River. If we just look at the numbers, you'll see that Robertson Stewart, in their study, which was published in '76, they recorded 129 species from the lower Congo. And remember, this is a short stretch of river. At the time, there were only about 600 species known from the entire Congo. So 129 from the lower Congo, a tiny little stretch, was interesting, and so something's going on. But perhaps, and think about it, actually in Europe, freshwater fishes of Europe are about 200 in all of Western Europe. So 129 in 300 kilometers is a lot. But more interesting in a way was that Robertson Stewart found that 34 of them were endemic. 34 of those species are only known from the lower Congo River. So there's something interesting, and that paper was the first to really point that out. And that's basically what we knew in 2006. Now, I am one to boast, but I don't mean to boast. Since our work began, we've done nothing but add to those numbers. Now, the lower Congo River, we found over 330 species there, and we found over 85 of them are endemic. So clearly, something very, very interesting is going on in this stretch of river. If we look very quickly, and I know it's a rather small slide, but if we look at the endemic species, so these are the species that presumably have evolved in situ in the lower Congo River. If we look at them, and there, as I say, we're over 85 and counting, you can see very quickly that whatever's happening seems to be happening and affecting all of the families of fishes, all of the types of fishes that occur in the lower Congo. They all are spinning off endemic species. So, you know, whether you're a catfish or a spiny eel or a lungfish, whatever you are, it seems that whatever's happening in the lower Congo River, it's affecting you. Now, some groups more than others, and George mentioned cichlids, and cichlids are these iconic fishes of evolutionary biology, and everyone's familiar with the extraordinary radiations of cichlid fishes in the Great Lakes of East and Central Africa. In the rivers, cichlids don't tend to speciate. That's a kind of orthodoxy that we've all grown up with. They speciate in lakes, but not in rivers. The lower Congo River is a real exception, and there are about 35 species of cichlids found in that tiny stretch of river, and 25 of them are endemic to that stretch of river. So something is going on. Now, you could say, and if we look at this next map, and what I've done here is divided Africa up into what are generally considered to be ichthyofaunal regions. So there's Lower Guinea, Congo, Kwanzaa. These are areas of Africa where you would find a certain kind of fishes. That geographical area, even if I didn't know where you'd been fishing, but if you showed me some fish, I could say, oh, you probably made those collections in the Kwanzaa. We can divide Africa up, the fishes of Africa up in this way. And look where Lower Congo is. It's, you know, we all know realtors tell us, location, location, location. If you're nestled between three big ichthyofaunal areas, you could readily imagine that the reason that the Lower Congo has got so many species in it doesn't explain in situ speciation, but why it's got so many fish species there might be because it's getting some from Lower Guinea, it's getting some from Kwanzaa, and it's getting some from Congo. So it's just its location that accounts for that incredible richness. But that clearly isn't the case, and I know everyone kind of hazes over when they see one of these phylogenetic trees, but we can use this, and this was just for cichlids, but it holds true for just about all of the fish groups that we've studied, is when you actually look phylogenetically at those endemic species from the lower Congo, and you find out who are they most closely related to, virtually every time their sister species is either found in the lower Congo or it's found in the Congo Basin generally. It's very rarely from anywhere else. So it really looks as if this area, the lower Congo River, has been almost entirely seeded from the diversification there, has been seeded from the Congo itself. So what is it? What is it about this short stretch of the Congo River that has turned it into this evolutionary hotspot of freshwater fish biodiversity? And we can look a little bit more closely at that lower Congo, so from the pool down to the Atlantic Ocean. And what we have above here is an elevational cross-section. And what you see immediately is that the pool Malabo, is at about 280 meters above sea level. The lower Congo River drops in elevation about 280 meters in elevation in a little more than 300 kilometers. An incredible, rapid gradational decline. And it does it stepwise and in a very interesting way. And the result, remember I said the Congo's draining, well, I hope I said it, 4 million square kilometers of Central Africa. It's draining that. All of that water is coming down and it's hitting Pool Malabo, and then it's falling over a sill, and then it's plunging down, literally plunging down from Pool Malabo down to the Atlantic. And how much water is it? Well, it's about 41,000 cubic meters of water a second. That is 3 times the flow of the Mississippi River. The Mississippi River flows over a great floodplain. Yes, we've constrained it and engineered it, but it's a big river with a big floodplain. All of that water, 3 times the flow of the Mississippi, is plunging down a gorge constrained in places less than half a kilometer across gorge. And the upshot is a lot of this. And I'm just going to quickly show you this video, which is not a very good video, but I'm going to let it run for a little bit. Maybe I can. So what you're looking at here, sorry, is the first rapid when the river flows out of Courmalevo. And I just want to point out for scale, that is a 6-foot guy there. And these rapids span the width of the Congo. It's incredible. Some of these rapids are beyond belief. Now, clearly, a system like this, there's tremendous potential for these series of rapids of this kind of dimension, that they clearly could have played an incredibly important role in the diversification of populations and of species in this system. I mean, you can imagine some species are adapted to a rapid, so they're going to hang out in the rapid. And then the still water between that rapid and the next rapid is going to be a barrier, right? Or if they're adapted to still water, the next rapid's going to be a barrier. So you can see how longitudinally these extreme hydrological structures, rapid, so-called calm stretch, rapid calm stretch, how that could really kind of help pump speciation out through simple allopatric speciation. And I'm going to talk about some of that in a minute. But before I do, I want to mention very briefly one of the first problems that we had to deal with. Basically, we knew there were a lot of rapids in the lower Congo River. But the problem was we didn't really know where the rapids were. We had maps, and there were some very good maps. Many Belgians lost their lives spending all of their careers trying to map the Congo Basin. And the maps are good. We have topographical maps, and we now have Landsat imagery. And they're very good, and they're very accurate. And they tell us a lot about landform and land cover. But they don't really tell us anything about what's happening in the water itself. It doesn't tell us, this map doesn't tell me where the rapids are. So one of the very first things we had to do was to work out, well, how are we gonna find out where all the rapids are? And until recently, to do that, we would have had to work directly via field work. But that's virtually impossible, because I told you that the lower Congo plunges through a gorge. So most of the habitation, most of the paths, most of the ways you get around in the lower Congo is a long way from the river itself. So access to the river is extremely difficult. And as you can imagine from seeing that video of the rapids, you can't really do it from the water. There's no way you could do that. So it's really problematical. So you're in this gorge, and even if you can get down there, it's extremely difficult to collect in this system. So clearly, we couldn't do our mapping by field work. So what we actually did was we utilized a way of looking at the nuances of radiation from some of these remotely sensed data sets that we got using object-oriented analysis. And we could really build up a picture of what was, a little bit more of what was going on in the water. And I just want to show you very quickly this, and I will whiz through it a bit because it's rather long. So many of you are very familiar with these kind of ways of looking at land cover, but you can actually do it looking at the cover in the actual form of the height of the water itself. So what you can see here in these blue areas are where we're mapping the calm stretches. And then you can see in the light blue, we're actually able to map where some of these rapid systems are. And here's a good example of one. So this whole system here would count as one rapid that we would be able to map. So that was the first thing we had to do. And we basically got a pretty good idea of where all the rapids actually were, these big hydrological structures that crossed the channel. And there they are. And perhaps not surprisingly, when we mapped up some of our species distributions, it was very, very clear that, yes, these rapids are forming barriers to some species, and speciation has clearly been it has been through allopatric separation of species. You know, you're in a rapid or you're not in a rapid. You're not going to move across still water to get to another rapid. So very clear, you know, it's a good story, and it makes sense. Rapids do present barriers to fishes of varying efficacy, and we do see longitudinal structuring of species down the lower Congo River. But we were also, at the same time, beginning to document extremely, surprisingly, low levels of migration of populations, individuals from populations within a species, up and down the Congo River, the lower Congo. And in this example, and I know it's a complicated slide, and I'm not going to dwell on the details, but in this case, we looked at various molecular markers. So we got individuals in the first case of this cichlid, Teleogramma depressum, and it only occurs in sites A, sites B, and site C. And we got populations from each of those locations. And we took DNA from their tissues. And we looked at various mitochondrial markers, and we looked at some anonymous nuclear markers. And we saw a lot of structure. And you can see that quite clearly in this case, the populations at site A are quite distinct from the populations at site B. So in this case, it looks that there's the break, and it looks very much as if these rapids here are probably separating the individuals from this species from here, and there's very little communication with the species just a little way down the river. We found a similar story with this guy. This is another cichlid, Lamprologus tigrapixilus, a little more complicated. This is a much more widespread animal, but a similar story, but this time, interestingly, the barrier seems to be at a different place. It's after the last large major rapids. You're separating populations from D and E from the rest of them. So really fine scale kind of stuff that you don't really expect on these sorts of, at this sort of level. So that was interesting. But then we also began to pick up cross-channel diversification. So in this case, we're back with our profile of the lower Congo River. Here's Pulmalebo. They're the first rapids after Pulmalebo, which is what I just showed you the video of. At this point here it is, we made collections of this species. We made collections on either side of the rapids, on either side of the river. And we're talking about a distance of less than 1.5 kilometers. When we did the same kind of analysis with these, we found that the population structuring here at an even finer geographical scale was even clearer. And in this case, we found that there was at least 5% sequence divergence between populations living here and populations living there, looking, to all intents and purposes, pretty much like the same thing. But obviously, they are separated. They're not crossing well, we can work out exactly how often they're crossing, and it's very, very few in a large number of generations makes it across the Congo River. So extreme example. I mean, this is like, when I give this talk in New York, it's like saying people in Manhattan are not going to be breeding with people in Hoboken. And I guess here you would say that, you know, people in Boston are not having anything to do with people in Cambridge because the Charles River, they got to get across it. I mean, it's at that kind of scale, which was very, it was very surprising. So clearly, something interesting is going on. You know, these very low levels of gene flow between populations that are geographically so close to each other. Very likely what we're looking at is the early processes of ecological speciation. You can imagine that these populations, either side of the rapids, they're adapting to local conditions, and over time, it's the classic evolutionary story, over time they're going to diverge. So it's kind of cool, but remember these are fish, not people. So we want to think about, well, how are the fish seeing the river? I mean, we look at it and we think, it's a piece of water, they can go across it, no problem. But clearly, the fish don't view the water in that way. So one thing that we really wanted to do to add to this picture was to understand what was actually going on in the water and look at it from a fish's perspective. Now, the very best way to do this is to utilize the pleiotropic effects of the Y chromosome. Because one of those is that they are crazy. And we managed to persuade a bunch of these, they were fabulous guys, a bunch of wonderful whitewater kayakers. And the hardest thing, that was not hard to say to them, go into that rapid. That was easy. It was hard to stop them going into the rapids. The hardest thing was to actually allow us to drill a hole in the bottom of their kayak so we could put a sound, an echo sounder so that we could measure depth. But they let us do it. So we put the echo sounders in, so we're measuring depth, and then we have this differential GPS, so we can locate exactly where they are in the river. And this is some of the stuff that they encountered, and I'll only do a bit of it.
Tripp and Scott crash head first into waves the size of school buses, each weighing about 30 tons.
Yeah, crazy. All right. Okay. So we were beginning to document strong structuring of populations, clearly mediated in some way by these extreme hydrological features in this river. But, particularly, I'm talking about the rapids. And we were finding up and down the river structuring of populations, and we were finding cross-channel where there were rapids, differentiation in species. So lots of structuring. But at the same time, we were beginning to get clues that something else was at play in this system. In places, we were beginning to pick up this signal of cross-channel diversification in fish populations in the absence of rapids. And that was really, really puzzling. Now, at one site in particular, and it's this place called Bulu, and it is marked here. So here's the pool. So Bulu is where the river, after expanding in this stretch, which, if you remember, I'm sorry, I should have pointed it out, when I showed you the locations of the rapids, there's a clustering of rapids down in this area, and another clustering of rapids here, but there's a stretch that doesn't look like there are any surface rapids. Anyway, so Bulu is just at the end of that place that seems to not have any surface rapids. And it's at a place where you get the first constriction of the channel, and the channel constricts down from about 1.5 kilometers across down to about 0.5 kilometer across. So there's a real constriction there. And at Bulu, a very strange fish had been found, and Roberts and Stewart had found it. Back in '76, they visited Bulu, and they found this cichlid fish, which I know you're not all cichlid lovers, nobody's perfect, but this is a very unusual cichlid. It's completely blind, and it's completely depigmented. Now, Roberts and Stewart managed to collect 2 specimens of this fish, and they tried and they tried and they tried. They even, after the expedition was over, they went back to Bulu to see if they could collect more, and they couldn't. So this fish, which his scientific name is Lamprologus lithops, was known only from those 2 specimens. So we figured, well, it was important. We need to get to Bulu. We need to get more of this fish, because to have a blind, depigmented cichlid is very cool. It could tell us a lot about evolution. So we went back and we searched and we searched. Our initial thought was, the animal's blind. It's got no eyes. It's a cave fish. We know that in cave habitats, you quite often get this series of losses of pigmentation, losses of vision, and all the rest of it. We scoured the place, no caves, no caves. We looked and we looked. We asked the local people, and I think I just have a little video which I'm going to skip because it's just us pulling into Bulu. We asked the local people if they knew the fish, and they said, sure, we know it. tastes disgusting. They call it Mondelli Bureau, which is very, very strange because Mondelli is the Congolese word for white person. And bureau is obviously French for office. So for some reason they called it the white man's office. And the only thing that I can think of is that I could just imagine Tyson Roberts, you know, coming to Bulu with a suitcase. Can't you? Can't you, with his briefcase and marching down the shore of the river? I don't know. But anyway, they said, yes, we know the fish. We call it Mondelli Bureau. Tastes disgusting. And we only find it dead. So we were like, what? They said, yeah, we occasionally will find one, you know, on the side of the river. And we were like, okay, really? Anyway, we stayed at that place. We looked and we looked. Unfortunately, many fish lost their lives as we searched for some of these blind cichlids, but we couldn't find any. But after a while of being there, some local fishermen came up with, am I in the right place? A couple of specimens, and they were dead, just as they'd said. They'd found them by the side of the river, dead, and there are two of them that I'm holding. Now, while we stayed and tried to unravel this mystery, they came to me with one of these Mondelli Bureau, one of these Leptologus lethops, that was not quite dead. It was the poor little thing. It actually died in my hand, but it was moribund. It was close to death, but it was alive. And as it died, we noticed something very interesting, and that was that its entire body was full of bubbles. I don't know if you can see that. I kind of outlined it just because you can't really see because the fish is so kind of translucent. But there were bubbles. There were air bubbles under its skin. There were air bubbles along its cheeks under its skin. There were air bubbles all over its gills. And that was the first time it just occurred to us, this guy looks like it's got the bends. It looks like it's decompressed. It looks like it's got what's called catastrophic. It was clearly, or it was very likely, living at depth and had somehow come up to the surface and had decompressed. Catastrophic decompression syndrome, which we do know about because that can be induced in fishes under various conditions. One of them would be you pull them up from deep sea with a fishing line very quickly. They will get the bends, they will degas, and they will die. So for the first time, because of this fish, we suddenly thought, well, maybe there's deep water here. And that's something never occurred to us. We just thought about rapids. So based on that, we were able to persuade some guys from the US Geological Survey and National Geographic to fund us deploying some quite sophisticated hydrological equipment. And that was, we really did deploy for the first time in a tropical system, acoustic Doppler current profilers. And I'm going to show you a little video of these just to give you a sense of what these things do. And one of the things that these things do, and again, we can have a depth trace, but at the same time, and we've got GPS, but what this thing is doing, and you're going to see it in a minute, not only can you make a depth profile, so we can actually see how deep it is, but we can also map the structure in the water, the velocity of the water. Now, this is done in marine systems quite a lot. It's not been done very much in freshwater systems, but because of the backscatter of all of the sediment in the water, we can actually visualize this. So that's what we did. And the system, the place I'm going to talk about is this, was at Bulu. So in Bulu, this is where the blind cichlid was. This is where we thought maybe there's deep water here. So just to quickly orient you, so here we have the curve, and on the black line showing you where we deployed the ADCP. And you can see along the bottom here, we've just got a bathymetry profile. And first of all, I'm going to show you the ADCP at this point. Now, this is the first bend in the river at Bulu. There were absolutely no surface rapids. The water looked completely normal. But when we actually, and I'm going to show you oops, I'm going to show you a trace. And here's a trace, just to orient you what's going on. You are standing in the middle of the water, of the riverbed, and you're looking downstream. So this is a cross-section. And what the color is coding, the color is giving you the streamwise velocity. And red water is flowing extremely fast downstream. But the blue water is flowing fast upstream. So we've got rivers within rivers. And we have a complete shear. I mean, there are no structures, water structures that span that channel. You have a complete shear here. So this bank is completely separated from that bank. If a little fish could swim out and it would hit the red, it would plunge it downstream. So for the first time, we were beginning to actually see why there was such structure for these fishes. I mean, you've got a river within a river. Remember, this is less than half a kilometer across, yet they might as well be 100 miles apart. Because for a fish to swim through this, a small little fish that likes to live on the banks and around rocks is not going to make it across. So it's quite clear that the river itself seems to be what's the barrier for these fishes, which is totally wacko, because we always think of water as being the passage for the fishes. But here, the water itself, the hydrology of this system, is actually separating populations of fishes. So that's kind of cool. Now, the next place we looked was at this point here, which is innocently labeled as a pool. Now, we couldn't go over the center of that region because, well, you're going to see why in a minute, but we were in 50-foot wooden pirogues, and we nearly lost them in this attempt. And this is what we were looking at here. First off, I hope you can see it, but look at the depth. This, now, we're talking about, at its deepest point, 165 meters, so about 540 feet deep. Tremendous depth. But now the colors of the water are not telling you how the water's going upstream and downstream. They're telling you how it's going up and down. So you have these tremendous vortices. This water is plunging upwards. This water is plunging down. And just to give you a little sense of the scale of it, these are kayaks. And this is one of these vortices. If this gets into this, it's going to get pulled down for about 500 feet to the bottom of that hole. I mean, it's incredible. These things we'd seen, but we didn't recognize what they were. We hadn't seen them from this perspective. When you're coming down this stretch of river, it looks like there's a hill in the middle of the river. And what that hill is the lip of this vortex. And they're standing structures. I mean, it's really absolutely extraordinary. And you can so imagine if a small fish living at extreme depth gets entrained in one of these vortices, you know, you're living on the bottom, you're kind of adapted to it, and then you see something to eat, and you maybe swim up a little bit too much, and then you get entrained in one of these, you come up to the surface, you catastrophically decompress, and you're dead. So it looks as if, you know, that's what's happening. That's the kind of little solving of the mystery. But the serendipity of collections, how making collections kind of then led on to, this is the deepest river in the world. It has canyons in it that are way below sea level. So remember the crazy kayakers, right? There he is, heading down for us. So let's look at what they got. So if we, now remember, these guys did not have the ADCP. They weren't measuring the velocity of water and looking at the complexity of the water column. They were just looking at the depth. Basically, they were just looking at the depth as they went downstream. And they went down a section of about 120 kilometers from the pool down to the beginning of that navigable stretch. So they did the first set of rapids. And here's the trace. And I want you to see this. 800 feet. This is not meters. We're not in the ocean. 800 feet. The hole, the canyon that we found at Bulu looks like a paddling pool compared to some of these things. So clearly an extraordinary system. And I will get back to a kind of conclusion with it. But I want to probably wind up very quickly. So I want to very briefly, and I really will do this very briefly, to talk just a little bit about how we, because, you know, I've collected thousands of fish. And I've got this very weird fish. I want to talk very, very quickly about some of the ways that we can use these specimens that we have in museum collections to actually start to begin to unravel some of the evolutionary questions that a system like this poses. Obviously, just having collections in and of themselves, alcoholic preserved specimens, is tremendously important. We need to know where life on the planet is, when it was where it was. We can look at it through time because of these collections. Just having the specimens is tremendously important. But there's a whole series of ways that we can use, and I'm going to be talking about fish, but this is probably true for most organisms. There are many ways we can deal with fish specimens. We can make skeletal preparations. Obviously, looking at bones can tell us some important big things about fish anatomy. And we've been doing this for centuries. We can do it in a much more refined way. We can use enzymes to clear away the body tissues, and then we can differentially stain with alizarin red to stain bones and alcium blue to stain cartilage. We can get very detailed anatomical specimens which can be dissected. So here taken off the jaws and the suspensorium and the pharynx. So we can do that. Of course, today we can also extract DNA from these specimens. And we can extract the DNA, and then we can amplify certain genes, and we can then use this data looking at the differences in sequence structure of different genes in numerous different individuals. And we can use that for the very basic baseline information of comparative biology and evolutionary biology. The first question, who's related to who? So to produce phylogenetic trees. We can do that. And I'm not going to go into the details of it, but here are our lithops, and it turns out that the sister to lithops is Lamprologus tigropictolus. So we now know who the sister is. That's very, very important information. So that's the baseline. And we can do that with DNA. It's easier to do it with DNA than with anatomy, but once we have that relationship, then we can use these anatomical features, in this case, this clearing and staining, and we can look in a little more detail at the kind of gross differences between these two species, which are each other's sister species, they're each other's sister, but they look so different from each other. And we can kind of look at that qualitatively, and we can see how the skull is extremely expanded. We could look at differences in swim bladders. We can also look at histology. We can make thin sections through these specimens, and we can stain them in ways that we can look at the cellular structure. And I know I'm running out of time, so I'm not going to dwell on it. But here's the first thing. Lamprologus lithops actually has eyes. And here they are. So this is lithops, and there's its sister. Its sister has perfectly normalized. Lithops has these tiny regressed eyes, which are situated under bone. We can look at very fine structure. We can, histology is a wonderful thing. We can look at the structure of that reduced lithops eye. We can even stain it using immunohistochemistry. We can actually see whether there are visual pigments in that eye. There's a tremendous amount of information you can get out of a museum specimen. We can do all that, and we can make all sorts of inferences. And one of the inferences is that lithops actually is cryptophthalmic. It has an eye. It's a very reduced eye. That eye is probably not capable of forming an image, but it probably is capable of detecting light and dark. So here we've got our first bit of information about the biology of this animal, which we can't really study in the field, because it would be easier to go to the bottom, to the Marianas Trench, than it would be go to the bottom of one of these trenches in the Congo River. But we can get a lot of information from a museum specimen. Now, I mean, years ago, CT scans were something that you had when you blew out your hip or something. Now we can CT scan, so X-ray computed tomography, CT scanning. We can do that with fishes, and we can get a tremendous amount of information. If lithops isn't seeing, and tigropictolus is seeing, Well, what about the brain structure? Is it compensating? Is it enhancing other sensory modalities? We can actually use these CT scans to reconstruct things like the brain. So again, new technologies are enabling us to do amazing things with museum specimens. And of course, today, in the age of genomics, we can go even further. And in fact, we're in the process. Because it's a cichlid, we have a beautifully well-annotated genome, not done by us, but by others. So we've got something to compare. Now we've got enough DNA from one of these dead specimens. And we know now who its sister species is. We know this one has no eyes. It has a hypertrophied craniofacial anatomy, or it's a cryptophthalmic eyes. It's lost pigment. We know what its sister species is. We can get their genomes. We have this comparative genome, so we can construct where every gene in their genomes are, and we can really start to begin to get at the real genetic underpinning of what's happening in this river, thousands of miles from here that we. Well, okay, enough hyperbole. So collections have become, they haven't become less important. Collections are not old-fashioned. Collections really are the key to the future. They're the most important things that we can pass down to next generations. We must support our collections, because we would never have known, I would even never have known when I started my work, the kinds of things, the kinds of data that we could get out of a preserved museum specimen. It's really, we can never second guess it. You know, what the value of these collections going forward, we have no idea. So we have to protect them. And as a plug for my institution, the American Museum, one of the things that we've done is we've started a new series. It's called Shelf Life. It's a really cool series talking about collections. And if you're interested, the title of this one that I did was 6 Ways to Prepare a Coelacanth. And all the comments were, you know, garlic sauce, lemon butter, yeah. No, it's all about, you know, the CT, the histology, and all of that. So there's the URL there. You can get it on YouTube, or you can get it through the museum. But it's a good series, and I should stop plugging it. Okay, so I really know that I've gone over my time probably. So I hope that I've given you a little idea of what a fabulous system the Lower Congo is. I haven't got time to go into all of this, but you know, I made a big deal about Lamprologus lethops and its sister species, Lamprologus tigropictolus, and they're here. But there are numerous other sister species pairs in different families over the tree of fish life that are doing exactly the same thing. These are trends that reoccur numerous times in the Lower Congo. It's such an extreme environment that selective pressure is huge, and fishes from completely unrelated as cows and gerbils are doing the same. You know, it's like you've got cows and gerbils looking exactly the same. That would be the equivalent in phylogenetic time of the difference between these fishes. So it's a fabulous system. And I'm going to really have to shut up. I'm not saying you're probably as exhausted as I am when I leave the Congo every year, but I'll skip that. And I'm going to skip through all of this because I just do not have time. Obama, we love him. Okay. And say thank you to all of you, but I want to acknowledge all of the people that have helped me in this work and continue to help me on this work. Those of which I'm most proud and to most indebted are my Congolese colleagues and my Congolese students. I can add, I'm happy to say, I don't have her picture yet, but I'm adding a young woman, Miriam Yoko. If she makes it through, she will be the first Congolese woman PhD, which will be really fabulous. So thanks so much to them. Thanks to all of the other people and funders and all the rest of it. This, you know, clearly it's not just me doing this. It's been a great collaborative journey. And of course, to all of the fish that gave their lives and are now in the collections at the museum. And I'll end with this. I call this my Mother Teresa shot. But it's me and my favorite place in the world and my accommodations for the next 3 weeks in Congo. So with that, I'm going to shut up and entertain any questions. I'm sorry I was so rushed. So.